So we are going to pick up where we left off. Um, Cell theory. You'll hear this a lot, especially if you are majoring in anything in the medical field or in science. Um, and this theory was not created by one person, as you will be learning. There was multiple people, and even still today, one of you could actually expand cell theory. The joys of science, and that's why you learn the scientific method. You can always prove something is wrong, or you can prove that it could be better. Um, Over the years, scientists have continued to use and improve microscopes, and they have discovered that all kinds of living things are made up of cells. In 1838, the German scientist named Slelden and Swan looked at all plants are made of cells, and they based that on conclusion of research on behalf of others. That was mainly they both looked into it, but it's more Slelden. Swan was more interested in animals, um, and humans and how they were made up of cells. And they had made important discoveries about living things. However, they didn't understand where cells came from until their until their time. Most people thought that living things could come from non-living matter. So literally, this is how education and science has evolved. People literally could think that people could fall from the skies or walk from the ocean, um, and be created, or walk out of fire, or come from a rock. Um, there was a lot of conspiracy theories that, thankfully, that these sciences looked into and researched and educated the population. Um, but in 1855, a German doctor um, named Randolph, and I'm not going to pronounce his name because I can't say it, proposed that new cells are formed only from existing cells, and all cells come from cells, he wrote. The observation and conclusions of Hook, um, Van Antonio van Leeuwenhoek, all of them with the help of Randolph and Slyden and Swan, created and developed the cell theory. And the cell theory is widely accepted explanation of the relationship between cells and living things. So in this part, hopefully that you wrote this down, um, that here are the two scientists that helped coalesce and start the foundation in the process of the cell theory. The cell theory is normally used in biology. Now, chemistry, you can talk about when you start talking about biochemistry, when you mix biology and chemistry together, and talk about different reactions and stuff like that. But there's no way you're going to talk about cells in physics, for real, or in Earth and space. You might talk about how different environments and forces could change things, but it doesn't really, it's not that part of science. So biology, or AKA life science, is where you're going to find the cell theory. Make sure you write all this down. This is the cell theory. All organisms are made up of cells. All living things are composed of cells. So all existing cells are produced by other cells, and the cell is the most basic unit of life. Um, there's also ones that word it a little bit different and say all cells are produced from other cells. Cells are the basic unit of life of structure and function in living things. But this one on the slide is what will be on your test, not the others. And the cell theory holds true for all living things, no matter how big or small. Since cells are common to all living things, they can provide evidence about all life because all cells come from other cells. Scientists can study cells to learn about the growth, um, the reproduction, and all the other functions that cells, all living things perform. So I'm going to give you about 2 to 3 minutes to write this down.
And guys, I want to welcome you back to a STEM Script Tutorial. We're going to talk about the cell theory, just about topic, but it's extremely important. Most people don't know that this theory is one of the most historic scientific, universally accepted concepts, and it essentially talks about how that living organisms on this planet are made up of cells, and scientists were able to discover these cells using very early microscopes. So I asked the student how to describe the cell theory, and they started talking about cell things. So I have to let you guys know, unfortunately, the cell theory has absolutely, positively nothing to do with cell. All right, let's get to it. A lot of good stuff to talk about. Hope you enjoy. Now, a lot of students ask, why is it important we learn about the cell theory? Now, it's not only one of the most significant scientific discoveries ever. We're talking about the discovery of the cell, but it also represented a major
So my bad. I fixed it. Apparently, it wasn't sharing the sound, so we're gonna start it over.
Back to a STEM stream tutorial. We're going to cover the cell theory today in this video in just about under 8 minutes. It's not too complicated of a topic, but it's extremely important. Most people don't know that this theory is one of the most historic, scientific, universally accepted concepts, and it essentially talks about how that living organisms on this planet are made up of cells. And scientists were able to discover these cells using very early microscopes. Now, I asked a student one time to describe the cell theory, and he started talking about cell phones. So I have to let you guys know, unfortunately, the cell theory has absolutely, positively nothing to do with cell phones. All right, let's get to it. A lot of good stuff to talk about. Hope you enjoy. Now, a lot of students ask, why is it important we learn about the cell theory? Now, it's not only one of the most significant scientific discoveries ever. We're talking about the discovery of the cell. But it also represented a major evolution in microscopes, microscope technology that allowed scientists to really visualize what they were hypothesizing in a laboratory. Now that they could actually see cells, they could prove that they existed. Now, these cells helped define the characteristics of life, but they did much more. They laid the groundwork for much of modern biology that you learn today in class and in labs. Those include microbiology and cell biology, as well as looking about common ancestry. Now, common ancestry is a field that we still are analyzing today, and there's a lot of companies that look at common ancestry, like 23andMe, that you may know of. It also contributed to the study of physiology and human disease. Now, the cell theory actually dates back to the 17th century with Robert Hooke. He was a man who worked for the Royal Society in England, and he was a very prominent member of the Royal Society. There was not too many pictures of him, but here's a really nice one we found by Rita Greer, painted in 2004. Now, as a member of the Royal Society, he was a philosopher, but he also did science experiments. And at the time, no one knew about cells. So he was actually the first one to discover cells, and he did so by using a very old-fashioned microscope that could only magnify up to 30 times magnification. And he analyzed oak tree bark and found that the oak tree bark had these little tiny compartments that he coined as cells. And the cell means small room. And he couldn't see inside these cells. He didn't know if they were alive, but he was the first one to discover that they existed. Now, shortly after Hooke published his results, a man by the name of Anton von Leeuwenhoek, and I know I'm butchering that name completely, I sincerely apologize, but he was much more skilled at building microscopes, and he was actually able to build one with 300 times the magnification, which impressed the hell out of everyone. He took protozoa and bacteria, which he didn't know were protozoa and bacteria at the time, but he named them animalcules, and he was able to see that they were modal. And he reported to the Royal Society in the 1600s that because these were modal organisms, and that represented a quality of life, that these specimens were living organisms. He's also credited for making the first observations of human red blood cells, and very importantly, sperm cells, because he was able to observe that sperm and egg were fusing together to create a new generation of cells. This was extremely important because it was the first evidence against spontaneous generation, that cells were not just arising out of nowhere, that they were coming from pre-existing cells that we know to be true today. Now, following the work of Hooke and Leeuwenhoek, who were the ones who really solidified that cells existed, the cell theory didn't really come into existence, the theory itself, until the mid-1800s. So scientists at the time were recognizing that cells existed, but the theory about what these cells represented and what they stood for in terms of life was just beginning to surface. So finally, in the mid-1800s, 3 German men by the name of Theodor Schwann, Matthias Jacob Schneid, and Rudolf Virchow, and I know if I'm butchering those names, again, I apologize, but these 3 men were finally able to come together with a large enough body of work and put together a concept known as the cell theory, which is actually a group of concepts we'll go into right now. But they are the ones who are credited for the cell theory, building upon the work of previous scientists like Hooke and Leeuwenhoek. All right, let's bring in the drum roll. Now, the cell theory is: number 1, all living organisms are composed of 1 or more cells. Number 2, the cell is the most basic unit of life. And number 3, all cells arise only from pre-existing cells. Now, my question for you guys is, which one of these components of the cell theory was the most controversial at the time? And the answer to that is number 3, that all cells arise only from pre-existing cells. And the reason that was so controversial is that it went against a very widely held belief that cells arose spontaneously. And it was also very difficult to prove. And it wasn't until experiments that were performed by Louis Pasteur where the scientific community was finally able to recognize that the theory of spontaneous generation was not correct and that cells arose only from pre-existing cells. Now, most people don't think of the cell theory when they think of Louis Pasteur. They immediately think of pasteurization, which is a widely used commercial technique that we implement to help protect our food. But he did indeed prove, or at least in most people's minds, convince that that spontaneous generation was incorrect using swan neck flask experiments where he boiled broth and proved that organisms must grow from pre-existing organisms. Now, interestingly, this topic of spontaneous generation is one that is still debated today amongst many prominent scientists in the scientific community. Now, the beauty of the cell theory is that it's constantly evolving as we continue to make more and more advancements in science and technology. Now into the 21st century, here are the few additions that the scientific community has made to the original cell theory and now into the modern cell theory. The first addition really represents research that scientists are now able to do on multicellular organisms, that they have now been able to truly appreciate that it is billions and trillions of cells working together to allow a very complex organism to function the way that it does. Energy flow is something that scientists are looking at a very molecular and micro level. The cells contain hereditary information. We know this to be true of DNA, which we will discuss in another video. That's an incredible topic to talk about. And also that cells in similar species have the same basic composition. I hope you guys at the end of this video have a much better understanding of why the cell theory was so important and where it originated from. It truly revolutionized the way the world thought about life, and it opened the doors and paved the way for so much more scientific research that continues on to this day. Just wanted to do a quick recap of what we discussed in the video. That the first being the cells were first discovered and observed in the 17th century by Robert Hooke and Anton von Leeuwenhoek. It was their work and discovery that cells existed that paved the way for the cell theory to come about in the mid-1800s. Now, the last component of the cell theory was very controversial at the time because it went directly against a widely held belief that cells arose spontaneously and did not come from pre-existing cells. Now, Louis Pasteur was the man who helped disprove that with his experiments, and this is actually a topic that's still being researched and debated today. And finally, the cell theory is continuing to evolve. It's continuing to be talked about and researched on, and that's the cool thing about science, that we're always going to be moving forward and trying to advance science and technology for the benefits of humanity. We appreciate you guys watching the video, and we'll see you next time on STEM Stream. Hey guys, I want to welcome you back.
So, Theodore Swan, I did not highlight this one. My bad. Watch, I'm about to fix this real quick. So, Theodore Swan, he was a physicist. Um, he also was the one who pretty much started and invented the understanding that animals had cells. Any type of animals had cells. Um, so that goes for us. Um, He also worked with Slyden, who was a botanist. And if you don't know what a botanist is, those are people that uh, study plants and other type of vegetations type stuff. Um, but Swan worked with a lot of different people. Um, he was a very, very interesting guy. Uh, all these people were. I'm just not going to say that at this time period during the Renaissance time, um, people work together and they try to come up with the best education solutions to stuff that they could. Um, you will be doing somewhat like a poster type thing. You will kind of like design an ad for one of the five scientists um, that we have talked about. And you will basically pick whoever you want, and you will be putting this person in the poster. Um, it is an easy grade. If you decide to pick Swan, you'll write three things about it, either from the slide or you can summarize it. I'm not expecting you to write everything down from the third bullet point or even from the last one. Uh, so I'll give you about 3 to 4 minutes just to write that one down. Um, So Slayton, he was born in 1804, he died in 1881. Um, he was a botanist, so he studied plants. Um, he wrote he wrote contributions to uh, Philogenos, which it was in 1838. We're definitely not going to get that because we are not diving into plants. Um, we will be talking about photosynthesis, but we are not diving all that way into plants. Um, he is contributed to looking under the microscope at the different structures of the plant cell, pretty much giving us that the animal cells have the same parts as plant cells. Only difference is the plants have a cell wall, uh, and the animal cells do not. He believed that the principle of biology was equal to and also important to the atomic theory of chemistry. Um, that is your periodic table, um, hydrogen, helium, all the different elements. Um, so he said that pretty much what we're doing is equal to or more important to what you were doing, which they were all important. But, you know, this is old school shade. Um, he also published how it's important, the importance of the cell nucleus, discovered in 1831 by Robert Brown, who's also a botanist and he was Scottish. So Scotland is close to Great Britain, so it is nowhere near Germany. So this is why these people traveled and constantly sent letters to each other on the work that they're doing. Um, one guy pretty much wrote letters all the time. Um, and he didn't write anything down. Uh, but biologists to accept Charles Darwin's theory of evolution, um, which was which we won't be talking about evolution also. We won't be diving deep into that. That's more biology. But a lot of scientists was not very accepting of the theory of evolution because they had their own beliefs how we was created and how the world started. Um, but Sladen finally, in his older days, he became a professor of botany in Russia in 1863.
Perplexed and Befuddled asks, can you do a show about making a microscope? Who names their kids Perplexed and Befuddled? Hey, to see the invisible world, your eyes need a little help. And even though squinting is wonderful and heavily endorsed on this channel, it doesn't quite do the job. But microscopes are just the help we need. They use curved glass to magnify light, which magnifies the world around us. To first understand microscopes, we need to know how light travels. Thousands of years ago, people began noticing some interesting properties of light. The first, now called the camera obscura, happens when light goes through a small opening. You can do this by blacking out a window and then making a tiny pinhole for light to come through. Magically, you will see the light outside the room project onto the opposite wall, only upside down. This happens because when light bounces off objects, like the sun's light bouncing off the person outside, the light bounces off in all directions, in straight lines. Remember, things look certain colors because those are the colors of photons that they bounce. If a person's head is above the hole in your blacked out window, the only way light bouncing off the head can enter the room is to angle downward, and makes the head look flipped. Same thing with photons that bounce up through the hole, and more importantly, this pinhole makes the person outside look magnified on the wall so that they seem bigger, just like a microscope. Curved glass, called lenses, can mimic this effect on light. When you shine light at a lens, like in a magnifying glass, the light gets angled into a pinhole, like in a camera obscura, called the focal point. From there, the image is flipped and gets larger. And this is how microscopes work. In most light microscopes, light shines up through your sample. Because of this, your sample needs to be really thin. Otherwise, light won't go through it. Looks like I'm not thin enough. I think I need to go on and eye it.
Wow.
When light goes through the sample, some colors of photons get absorbed by the object. The rest bounce straight through towards the first microscope lens. Once inside the tube, the lens angles the light to its focal point and then magnifies the flipped image. The last lens at the top, called the eyepiece, takes that flipped magnified image and magnifies it again. Hot and ready for that eye to see. Simple microscopes like a foldscope skip that eyepiece and tube altogether and just use one lens. A glass ball works great for this. With light coming up through your object and hitting that glass ball, the image gets flipped and magnified and you get a microscope. You can build your own simple microscope using a drop of water. Instructions are in the link below, but by hole punching a clear plastic container and letting water fill in the hole, you create a lens that magnifies light. Then all you need is light that can shine up through your sample and voila! A homemade microscope that cost $0. So stop naming kids perplexed or befuddled, please. Keep asking questions and keep on squinting. Did you know we have a website full of science activities you can do? PlanetSquint.com. On it you can find how to make your own microscope as well as other science goodness. So check it out.
Rudolph. He was born in 1821. He died in 1902. He was a physician and he was an anthropologist. He used, which was very creative. He was more into, he championed social reforms, and they contribute to the development of anthropology as a modern science. And when we talk about anthropology, you can ask Mr. Vincent more about it, because it's not in my jurisdiction. because anthropology is more of the study of origin and development of human society and cultures. Um, so it's going way, way, way back. And Mr. Vincent can definitely explain it more to you than I can. Um, he also used the modern process by his application of his part of cell theory to explain effects of diseases in the organs and in the tissues of your body. So he was looking at irregular cells, uh, which we will be talking about some of, which most of the time they cause cancer. Um, they can cause cancer to spread, or they can be malignant to where it's not harmful to a certain extent. Um, and he also published a book, well, an article review in 1845. It was more of paper, and he was talking about the two earliest reports caused cases of leukemia. Leukemia is part of cancer, and he was looking at those different parts of the cells that didn't form correctly and cause problems.
He was a political activist, archaeologist, doctor, publicist, pioneer, for sure a workaholic, father of modern pathology, and the grandfather of nowadays hipster. Rudolf Virchow. Born on the 13th of October in 1821 in Schivelbein, in the rural Polish province of Pommern, he soon realized that Berlin was, and still is, the place to be. In 1839, he started his medical education at the Pepinière Military Medical Academy. He asked his parents for money to keep up with the latest Berlin Fantasy-Geschmack. Only a year after graduating from medical school, he had such an abundance of revolutionary ideas, he decided to found his own journals as an outlet. The Archive for Pathological Anatomy and Physiology, nowadays short Virchow Archives, are one of the oldest medical journals still published today. As a born revolutionist and fighter, he fought on the side of the Democrats in the 1848 March Revolution in Berlin, being part of the Democratic Congress. Consequently, losing his new position at the Charité Hospital, he swore to abstain from further political activities, and was hired as a professor in Germany's first Institute for Pathological Anatomy in 1849. In Würzburg, he not only found scientific fulfillment, but also the love of his life and children, and more children. Passion drives performance. Being held from further political revolutions, he turned to medical revolutions instead, overturning old ideas of humoral origin of disease. That's bullshit. His best friend at this time? His microscope. After 7 long years in Würzburg, in 1856, Berlin finally was ready for Virchow and invited him back as the chair of the newly established Institute of Pathological Anatomy and Physiology at the Charité. In 1858, he published the seminal work Cellular Pathology, the Bible of pathology and the foundation of cellular biology as we know it. Omnis cellula e cellula. In his work, the Pope of Pathology also described the trinity underlying all of our understanding of thrombosis and embolism, the Virchow trias, giving jobs to thousands of strokologists, cardiologists, and hematologists around the globe today. He precisely described diseases and coined terms like leukemia, chordoma, parenchyma, and amyloid degeneration. His true love, however, always remained with the individual cells, describing new ones like the microglia and finding the cellular origin of diseases. In his eye, politics was nothing but cellular pathology on the bigger scale, so, technically, he was not breaking his oath when he started climbing the political career ladder and founding the liberal Deutsche Fortschrittspartei. Progressive ideas, even up to our standards today, we need public health care, soon attracted the rage of the political elites again. In the Great Sausage Duel of 1865, Otto von Bismarck, the president of Prussia, was so fed up with Virchow, he challenged him to a duel to the death. It's either you or me. Virchow cleverly used this to make his point of the importance of hygiene and meat inspection, choosing Trichinella-infected sausages as the weapon. With Virchow's help, Berlin became one of the first European cities to get canalization and central water supply. Whenever not busy revolutionizing the way medicine describes diseases or fighting for higher social standards and better working conditions, he liked to travel. And because he collected a whole lot of stuff on the way and his archive for pathological samples was overflowing, he decided to put them elsewhere. Where else but a museum? 2 of them, one for ethnology, one for pathology. Virchow, many concluded, can't be the name of a single person, but must be the term describing a collective of workaholics living around the turn of the century.
Okay. So, in either in the back of the room or on Mr. Ward's desk, there will be blank sheets of paper. Make sure you put your name on it first and last. I'm not going to be doing a guessing game who is what paper. So if you really want to grade, you put your name on it first and last, and your class period. pick one of the 5 scientists that you want to create an ad for. You can color it, you don't have to color it. Just make sure it has their name, somewhat creative, and 3 things about that person. Now, if you go over and you do more than 3 things, I will definitely, if they are correct, then I will give you extra points for it. If you do not get it done in class, take it home. I'll give you till Friday. If it's not turned in by Friday, then I won't take it. So you have literally almost 2 days or more. So get it done, turn it in. Tomorrow we will go over 8 slides about different parts of the cells. Friday we will do the last 8 of the slides. And then on Monday you will get your lab partners and you will sit at your table that you are assigned to with your lab partner and you will start your stuff. Which will be life in the cell. One day will be plant, one day will be animal cell. Creating a cell. There's just a whole bunch of stuff you can do. So get it done and turn it in by Friday.